Hi, let's move on to synapses now. Synapses, if you recall from anatomy, it could be electrical. When we talked about the heart, we talked about gap junctions. It was one of those structures that anatomy was so hard to find, which were the intercalated discs, right? And what those are these connections between cells. And the heart is different in that an electrical signal can pass directly from one cell to another without releasing a neurotransmitter. So gap junctions are special for that. Most synapses don't work that way in the body. Most of them are chemical, and they will use a neurotransmitter. So in the diagram below here, you have the terminal end here of an axon and the next cell coming along. And you would have to have the release of a neurotransmitter that comes into the synaptic cleft that then attaches onto the channel on the next cell, and then that action potential can continue moving on. So we're assuming we have an action potential traveling down here. It can't just jump over like it would in a gap junction. It would need the release of the neurotransmitter and then again opening of sodium channels after that neurotransmitter has attached onto that receptor. So very different the way that functions. And most of them, as I said, will be chemical. So let's think of those chemical connections. Think about, is that chemical message that's sent from cell 1 to cell 2 or presynaptic cell to postsynaptic cell, is it always stimulatory? Is it always sending a signal saying, go? Well, it turns out that no. They could be stimulatory or they could be inhibitory. So in my picture here, you have someone lifting weights, sort of doing a curl. And once you do that, you would have to come back down again with your forearm. And in the coming down of your forearm, you're going to have certain muscles that are being inhibited for you to be able to move that action, for your triceps to actually engage. And stimulatory, it's going to be stimulatory for the triceps and inhibitory for the biceps when you're bringing it back down again. So I want you to see how important it is that both of those are functioning in muscles for you to be able to have very smooth movements in your muscles. You're always going to have to be contracting one and holding another one in place, another muscle in place. So it's important because of this dampening of muscle action. A very interesting example of when that doesn't work is in Parkinson's disease. You don't have the dampening of muscle action in the hand, and then a hand tremor develops. So the tremor is there because you don't have the inhibitory response at the level of that muscle, sort of calming it down. The question here is, how are excitatory or inhibitory signals named? So you'll have to know this. Excitatory postsynaptic potential is a EPSP. So it's excitatory, it's postsynaptic because it's the cell that comes after that connection. And that's where the potential is going to happen. So EPSP, it's going to trigger an action potential, or the inhibitory postsynaptic potential, or IPSPs. And those are the ones that are contributing to the blocking of an action, the tremor in the hand that you don't want. When you bring your weight down, you don't want to be moving it around in all directions. You want it to be a smooth action. And that's because you have inhibitory postsynaptic potentials that are also being part of that movement. Now, think about, can both of these be happening on the same neuron? Could you actually get a message coming to a neuron saying, oh, you better excite that muscle, and another message coming saying, oh, don't you do that, why don't you stop that excitation? So can both of those stimuli be occurring on the same neuron? And this picture here is one cell body is going to have the input of lots of other terminal axons here. This is the terminal point of those axons. So it could be receiving a lot of messages. Do all those messages have to be excitatory or inhibitory, or could you have a mixture? Well, you could have a mixture. So one neuron has so many of those signals that some could be inhibitory and some could be excitatory. Well, who would win out? Well, it's kind of a tug of war. If most of them are inhibitory, it will be inhibited. If most of them are excitatory, it will be excited, and it will send a message. So let's look at that. My little stick men now are duking it out. They're fighting it out because that's what's going to determine that action potential. Do we have one? Yes, because it's more excitatory, or no, because it's inhibitory. That is called summation, when you add together the graded potentials of those incoming signals. So to sum is to add. You're adding together those incoming signals. If the overall membrane potential is above threshold, the action potential is produced, correct? Remember that lowest line? If that membrane potential reaches that minus 55 millivolts, it's going to fire, and you're going to have an action potential going on there. So what kind of summation exists? Well, we could have spatial summation, in which case you're noticing that this incoming is happening in different places, in location A or B or C here. So it's in space, a different space on the cell body, a different space. This one's coming in here, and this one's going to come in here. And let's say these are all presynaptic. And let's say that they are all stimulatory. My little stick people now are helping each other out. So this is the stimulation of A plus B plus C, you're summing them together, are enough for them to add up and reach this axon hillock, and yes, a potential will fire. You'll have an action potential that's firing. So that is spatial summation, synapses on different locations of that same cell body. What if not all of them are summing? What if one of them is inhibitory? So in this case, we can make this A here is inhibitory, and we'll make this B, and we'll make this C. So the inhibitory is going to actually annul some of that current that's coming in from B and from C, and it does it by making it hyperpolarized. So it makes it much harder to depolarize that membrane, because it makes it, instead of minus 70 to start with, minus 90. So it has to do with charge, and it's going to make it harder for it to reach threshold. And perhaps, in this case, they're duking it out here, little stick guys, and maybe no action potential, because not enough of that depolarization occurred in the cell body in order for it to reach the axon hillock. What about a different kind of summation, a temporal summation? In this case, you could have the synapse be in the same place, but happening quickly. So you fire, and it's happening so quickly that there's going to be an added response and an added depolarization. So let's look at what that would look like in a graph. You have a membrane potential here over time. And let's say you have a small little incoming message. Nothing much happens because it does not reach threshold. And then you wait a while, and then you get another little message. But still, not enough to reach threshold, because this is far from threshold here. Now we're going to change the game. We're going to have that summation happening more quickly. So if we delivered that impulse at x1 in time in milliseconds, and this far apart at x2, we're going to bring those together so they can happen more quickly. So boom, boom, more quickly. And so we begin a depolarization of A1, and we don't have time for it to return down, and we depolarize it again. That summation that's happening there of that current is going to reach the 55, and you're going to have the development of a full-grown action potential. So my stick guys here are helping each other out, one over the other one. So that has to do in a summation in time, not in space. This is more pictures of a spatial summation where you have inhibitory and then a couple of excitatory indicated by the plus sign. And then a membrane potential that will or will not fire depending on whether this inhibition was large enough or not. Summation in time or space. Here we have first delivery of that charge, and then here we've delivered it much more quickly one after the other, and it has reached 55 millivolts and fired. This is showing spatial summation where you have a small depolarization doesn't reach threshold, another small depolarization doesn't reach threshold, but then if you actually summed one in one place and one in the other place together in different locations, they would add up enough to have an action potential. Here it's showing a cancellation, and the cancellation, remember I told you, is we're assuming we're starting at -70. All this is starting at -70. Well, what if you had some ion that made it even more negative in the cell to start with? You would be starting below -70, and it makes it harder to reach this threshold because that distance has increased. Chlorine tends to do that. It's negative. If we bring negative chlorine into the cell, it's going to go from -70 to -75, -80, depends how much chlorine enters the cell. So that's a good little summarization of what's happening. It's just an enlarged slide for you to look at. There also could be a presynaptic inhibition. And in this case, an inhibitory neuron synapses on a collateral of the presynaptic neuron. So here we are, an excitatory neuron. It's reached threshold at the axon hillock. It travels, and it's reaching a response. It's going to release neurotransmitter. We're doing really well. But over here in this branch here, there's an inhibitory neuron. And this presynaptic terminal that was going to communicate with this cell is now blocked. So that's another form of inhibition where the rest of the neuron is allowing cells to fire except for one particular location is blocked and there is no response. Postsynaptic inhibition, all targets of the postsynaptic neuron are inhibited equally. So here, if I had, this is confusing because it looks red, but it doesn't mean that it's not stimulating it. It's a positive. And here it's a negative, not enough to reach. It won't, none of these are going to respond because this was this postsynaptic inhibition. How does this work? How do you get one to be excitatory and one to be inhibitory? So the excitatory postsynaptic opens acetylcholine channels. Excuse me, let me rephrase that. The acetylcholine opens the sodium channel. The inhibitory postsynaptic potential, the GABA, opens chlorine channels. And chlorine is negative, and it will bring down that resting membrane potential. And that's where we stop.